Check, check, check. All right, class, let's continue on with our stages of the adaptive immune system, our third line of defense. Let's talk about cell-mediated immunity. Cell-mediated immunity, aka CMI, not TMI, not too much information, but CMI, cell-mediated immunity, is going to be responsible, or primarily going to be responsible, or defined as T cells destroying other human cells. So when a T cell directly attacks a host cell, that's cell-mediated immunity. When we focused on our B cells, we focused on our humoral or blood immunity. Our T cell reactions are very complex and diverse. There's a lot of T cells out there, and most textbooks, this one included, don't necessarily do it justice. We have lots of subtypes of our T cells. The actions of our T cells are always going to be dictated by our antigen-presenting cells, or a gamma delta T cell. We first are going to need to be able to activate the T cell, and primarily we activate them with those antigen-presenting cells. If we look at some of our major histocompatibility complexes or self-recognition markers, they are going to be restricted. So if we have a human cell that just has MHCs present on it, those MHCs are a way of saying, hey, I belong in the body, please don't destroy me. And generally speaking, T cells are going to be pre-programmed to ignore human cells that present those MHCs, those self-markers. Our T cells that are activated are going to start producing cytokines. Cytokines are a specific category of chemical that cause other cells to be destroyed. Our T cells, once they're stimulated, can then activate other T cells, or B cells, or phagocytes in general. Particularly if we activate the T helper cell, the rest of these cell types, the T cytotoxic, the T memory, the B cells, and the phagocytes will all then subsequently be activated. So if we look at T helper cell is a very important one. That T helper cell comes in a variety of different flavors. They, generally speaking, activate other cells of the adaptive immune system. And on this figure, there's different T helper cell subtypes. T helper cell 1, T cell 2, 17. Apparently your textbook decided to skip numbers 3 through 16. What I want you to take away from the slide is all of these T helper cells have CD4 as their cluster of differentiation proteins on their surface. Our T regulatory cells, or TRs, also have CD4, but our cytotoxic T cells, or Cs, have CD8. CD8 is unique to cytotoxic T cells, T cells that destroy other infected cells. CD4 will be present on the other T cell types. If we look at those T helper cells, they all have CD4. Those T cells are going to activate macrophages. The T helper cells activate macrophages. Macrophages are one of the phagocytes. They're also going to release some chemicals. And for the purposes of this class and for this test, I do not want you to memorize or focus on the different kinds of interleukins or interferon gammas. What I want you to focus on is that different kinds of helper cells release different kinds of chemicals to trigger different kinds of events. So our T helper cells can release some chemicals to recruit macrophages. Our T helper cells can release some chemicals that cause the growth of more T cells, including cytotoxic T cells, and our T helper cells can release some chemicals. Again, don't focus on the chemical names, and those chemicals can stimulate B cell differentiation. T helper cells are the single most common T cell type. They make up the majority of the T cell population, coming in at 65%. Our target cells that are going to focus on our T cytotoxic cells, or cytotoxic T cells, T sub C, can destroy human cells. So if we look at any cell that has an intracellular pathogen, typically a virus, that's going to be targeted by a cytotoxic T cell. A cancerous cell is targeted by a cytotoxic T cell, or if we have a tissue transplant from another animal or from another human, oftentimes our cytotoxic T cells will start to destroy those organs and tissues that are transplanted into our bodies one cell at a time. Our gamma delta T cells are the new kids on the block. This is one of the new additions, or new, one of the new cell types that was added to this edition of the textbook as compared to the prior edition of the textbook. Our gamma delta T cells aren't very well understood. They are distinct from the other T cell types. Gamma delta T cells can be activated directly by an antigen. Our gamma delta T cells can respond directly to a PAMP, which is the same way that our non-specific white blood cells of the stage 2 of immunity also respond or can be triggered. Since our gamma delta T cells don't require or do not require the antigen presenting cells, they can respond quickly. Another unique feature of our gamma delta T cells is that they can produce T memory cells. Just like our B cells can produce B memory cells that give us the ability to rapidly produce antibodies for extracellular pathogens, our gamma delta T cells produce T memory cells, which give us the ability to rapidly proliferate and reproduce our T cells when we are being exposed to intracellular pathogens. Since these gamma delta T cells do not require an antigen presenting cell, and they can help to activate the specific immune system or the acquired immune system, they are going to be a bridge cell type. They help to connect stage 2 to stage 3 of immunity, our second line of defense to our third line of defense. We also have natural killer cells, and there's two flavors of natural killer cells. The first flavor, just the NK, is strictly going to be non-specific and is considered a second line of defense. It doesn't have any specificity for antigens. It patrols the body through the spleen, blood, and lungs, and other tissues of the body, and it will attack cancerous and virus cells. Whenever a cell is infected with the virus, there are some generic signals or red flags that cell can put up to say, hey, I should be destroyed. And those generic red flags the cell puts on its surface are independent of the specific kind of virus that's infecting it. So our natural killer cells can kill cells in almost the same way that a cytotoxic T cell can. I should emphasize again that our natural killer cells are not part of our specific immune system or stage 3, our third line of defense. They are part of our non-specific immune system. But then we also have natural killer T cells, NKT cells. These natural killer T cells are hybrids between the natural killer cells and cytotoxic T cells. These natural killer T cells will have T cell receptors for a specific antigen and release very large amount of cytokines very quickly. Because they have the ability to respond to specific antigens and also to generic antigens, these are going to be considered another bridge cell type between nonspecific and specific immunity. The other bridge cell type that we talked about was the gamma delta T cell. So our two bridge cell types are gamma delta T cells and natural killer cells, natural killer T cells. Here's a figure from your text illustrating the different cell types. If we activate a generic T cell that has a CD4 receptor, it can turn into a T memory cell. It can turn into a variety of T helper cells. And that variety of T helper cells will activate B cells or will recruit macrophages. Activated B cells are then going to turn into B memory cells, plasma cells, or B regulatory cells. And those B regulatory cells then go back and interact with the T helper cells to establish some give and take with positive and negative feedback loops in play. If we take this T helper cell and expose it to an antigen presenting cell, and we activate the CD8 complex, we can then have a cytotoxic T cell produce. So we can then have a memory T cell for cytotoxicity, a CD8 memory T cell, and then the cytotoxically active T cell will then destroy human cells or host cells that have intracellular pathogens and cause them to be lysed. Oops, did I skip a slide? No, I didn't. Okay, so concept check class. Cytotoxic T cells are programmed to kill all of the following except for A, cancer cells, B, virus-infected cells, C, bacteria, D, foreign cells grafted into the body, or E, none of the choices is correct. Please rewind this video to find the answer, or you can flip back in your notes, flip back in your PowerPoints, pause it, and get me an answer. 1, 2, 3, 4, 5. Cytotoxic T cells can destroy cancerous cells, they can destroy virus-infected cells, and they can destroy foreign cells that are grafted into the body, but they do not destroy bacteria. Bacteria are primarily going to be eliminated when antibodies from plasma cells bind to those bacterium, and then that bacterium covered in antibodies is phagocytized by some of our phagocytotically active cells, such as the macrophage or the neutrophils. If you have any questions regarding this material, please post them in the discussion board or shoot me an email. Happy studies!